Good morning, students. In yesterday class, we studied about the C4 cycle and CAM pathway. After that, today we are going to learn one more important topic, that is the photorespiration. Okay, the topic was photorespiration. This photorespiration were also called as glycolate cycle, or C2 cycle, or photosynthetic carbon oxidation PCO, photosynthetic carbon oxidation. Okay. So, the photorespiration where we can observe. In our previous class we discussed about the C3 cycle in C3 plants. Okay. In C4 plants also we find C3 pathway. Okay. So, in this, in which type of plants the photorespiration we can observe? Especially in C3 plants, this photorespiration occurs at high oxygen concentrations and at high light intensities. Okay, only C3 plants shows photorespiration mechanism. The C4 plants does not show any photorespiration mechanism. Already we know why the C4 plants does not show photorespiration mechanism. Okay. So, before we start the photorespiration mechanism, we have to know about the Rubisco enzyme once again. Rubisco. Okay. At the cycle of C3, already we discussed about the Rubisco enzyme role. Okay. This Rubisco enzyme plays a dual role. Okay. This Rubisco enzyme plays a dual role. That is RUBP carboxylase, oxygenase. Okay, this is the dual role of Rubisco. When the CO2 concentration is sufficient, it is involved in the C3 cycle. When oxygen concentration is more, at high light intensities, then what happen actually, instead of C3 cycle, it will be involved in another pathway, that is the photorespiration mechanism. That means here RUBP binds with oxygen. Okay, in C3 plants the RUBP binds with CO2, especially in C3 cycle. But at high light intensities, if the oxygen concentration is more compared with the CO2, this RUBP binds with the oxygen. Okay. So, whenever this RUBP binds with the oxygen, there is another process, that is the photorespiration mechanism. Actually, this photorespiration mechanism is an wasteful process. It is an wasteful process. There is no formation of ATP and NADPH2. Again, there is an utilization of ATP in this pathway. Utilization of ATP in this pathway, then 25% of the carbon loss by this pathway. So, that is why it is an wasteful process, especially in C3 plants. In C4 plants, the photorespiration is completely absent, because of in C4 plants there are two type of cells, mesophyll and bundle sheath cells. In bundle sheath cells, what happens actually? Malic acid undergoes decarboxylation. Okay, there is a continuous decarboxylation, because of there is a high amount of carbon dioxide release at the region of Rubisco. So, that is why there is no problem, because of the high amount of CO2, there is a continuation of C3 cycle in C4 plants. So, there is no oxygen concentration actually. So, that is why the photorespiration is completely absent in C4 plants. Okay. But in C3 plant we find only one type of cells. At the time of oxygen concentration only, the mechanism what we observed actually, that is photorespiration mechanism. Okay. So, in this photorespiration mechanism, photorespiration, uptake of oxygen in the presence of light. Okay, just you see in the word there is a meaning, uptake of oxygen and release of CO2 in the presence of light, just we call as photorespiration. Okay, here uptake of oxygen and release of carbon dioxide in the presence of light, simply it is called as photorespiration process. Okay. So, this photorespiration mechanism was first discovered by Decker and Teo. Decker and Teo in 1959. In the year of 1959, this photorespiration was discovered by Decker and Teo in tobacco plants. In tobacco plants. Okay. What they observed actually, at the high amount of oxygen rate, okay, at the high amount of oxygen or high concentration of oxygen, the photosynthesis will be inhibited. In the presence of light, the inhibition of photosynthesis at high concentration of oxygen were also called as Warburg effect. We also call as Warburg effect. Okay, the inhibition of photosynthesis at high concentration of oxygen, the presence of light, it is called as Warburg effect. Okay. So, now in this photorespiration, actually there is an involvement of 3 types of cell organelles. Okay. In this photorespiration mechanism, there is an involvement of 3 types of cell organelles. So, simply you remember the party, that is CPM party. Okay. In this CPM party, we find 3 types of cell organelles. So, C means chloroplast. C means chloroplast. P means peroxisome. Peroxisome. M means mitochondria. Mitochondria. Okay. Photorespiration in a process, wasteful process అయినప్పటికీ ఎన్ని సెల్ ఆర్గనెల్స్లో ఇది జరుగుతుంది? 3 సెల్ ఆర్గనెల్స్లో. 1 chloroplast, second one peroxisome, third one mitochondria. Okay. So, in this photorespiration mechanism, in these 3 type of cell organelles, there is a loss of carbon dioxide and utilization of ATP and utilization of oxygen. Just you see. In this, first we have to take chloroplast. In the chloroplast, 2 molecules of RUBP, 2 molecules of RUBP combined with the or associated with the 2 molecules of oxygen. Okay. So, not CO2, this is the oxygen. Actually, RUBP binds with the CO2, then only we find the C3 cycle. But here, is 2 molecules of RUBP binds with the 2 molecules of oxygen. Then what is the result of this combination? Then there is a formation of 2 molecules of PGA, 2 molecules of phosphoglycolates. Phosphoglycolates. In the case of C3 cycle, we will get only PGA, that is the phosphoglyceric acid, is the first stable product. But here what you observed actually, when RUBP binds with the oxygen, then there is a formation of 2 types of products, that is 2 molecules of phosphoglyceric acid, 2 molecules of phosphoglycolates. So, this is a 3 carbon compound, this is a 2 carbon compound. Okay. So, now you see this is a 5 carbon compound. Okay, then 2 molecules of PGA and 2 molecules of phosphoglycolates will get as a post products. Okay. Immediately the phosphoglycolate, just you keep PGA. This phosphoglycolate undergoes dephosphorylation in the presence of phosphatase enzyme. Okay. This phosphoglycolate immediately undergoes just dephosphorylation. Just you see, in the presence of phosphatase enzyme, we will get the 2 molecules of glycolates. This is the 2 carbon compound, 2 molecules of glycolates. So, in this 2 molecules of glycolate is the stable product. So, that is why this cycle we are also called as glycolate cycle. We are also called as glycolate cycle. Okay. So, this two steps occurs in chloroplast. This two steps occurs in where? Chloroplast. Okay. So this is the 2 carbon compound. That's why this photorespiration we are also called as C2 cycle. In this photorespiration mechanism, there is a photosynthetic carbon oxidation. So that's why it is called as PCO. Okay. So now after the formation of glycolates, this glycolate diffused into another cell organelle, that is the peroxisome. Okay, that is the peroxisome. So these two molecules of glycolates, undergoes oxygenation. Okay, in this two molecules of glycolates, in the presence of glycolic acid oxidase, it goes to oxygenation. Then we will get the two molecules of glyoxylate and hydrogen peroxide. Okay, there is another oxygenation. These two molecules of glycolate binds with the two molecules of oxygen in the presence of glycolic acid oxidase. Then we will get the two molecules of glyoxylate and hydrogen peroxide. This hydrogen peroxide dissociates into water and oxygen in the presence of catalase enzyme. Okay. So then these two molecules of glyoxylate, undergoes transamination reaction. Okay, undergoes transamination reaction, just your amino group will be added. Transamination reaction in the presence of transaminase enzyme. Then we will get the two molecules of glycine. It is a simple amino acid. Okay, two molecules of glycine. So these two steps occurs in peroxisome. Okay, after the formation of glycolate, then there is an oxygenation. It forms glyoxylate and H2O2. After that, this glyoxylate undergoes transamination reaction. Then we will get the two molecules of glycine. Okay, these are the 2 carbon compound, 2 molecules of glycine. Now, after the formation of glycine, this glycine diffuses into another cell organelle, that is the mitochondria. Okay, this glycine diffuses into mitochondria. So whenever it reaches mitochondria, this 2 molecules of glycine undergoes oxidative decarboxylation. So then there is a release of CO2. This reaction undergoes oxidation, then NAD plus converts to NADH plus H plus. Okay. So in the mitochondria, what happens actually? There is an oxidative decarboxylation mechanism. So in this oxidative decarboxylation, there is a release of CO2, that means loss of CO2. If there is 100% of CO2 in these components, there is a loss of CO2, that means 25% of the CO2 loss by this process. Okay. Remaining carbon dioxide will be involved in photorespiration mechanism, at last form the phosphoglyceric acid. Okay. The loss of CO2 occurs in mitochondria, that is by oxidative decarboxylation mechanism. Here also there is a release of ammonia, it will be involved some other metabolic activities. Okay, now after oxidative decarboxylation we will get the serine, only one molecule of serine. Yes, you see this is the 2 molecules of glycine, 2 carbon compound, 2 into 2, 4 carbon compounds. But here what happens, one carbon will be removed in the form of CO2. So then only 3 carbon compound will be formed, that is 1 molecule of serine. Okay, that's why we have to take 2 RUBP, 2 O2. Okay. So this 1 molecule of serine is the 3 carbon compound actually. So this serine again transported into peroxisome. Okay, this serine again transported into peroxisome. In peroxisome, this serine, okay, undergoes deamination. This serine undergoes deamination. This amino group reached to this area. Okay, this amino group was fulfilled in this. This serine undergoes deamination, then we will get the hydroxypyruvate. Then we will get the hydroxypyruvate. Okay. So, after the formation of hydroxypyruvate, it undergoes reduction. Here oxidation, now here reduction. The NADH plus H plus will be utilized, it converts to NADP plus, this will be balanced. Okay. So, now this hydroxypyruvate converts to glycerate by the process of reduction. This glycerate transports or diffuses into chloroplast and utilized ATP. After utilization of ATP, again we will get the PGA. This PGA and this PGA will be involved in the Calvin cycle again. Okay. So, this is the photorespiration mechanism. What you observed actually, there is an utilization of oxygen. Okay. There is a formation of phosphoglycolates, then loss of CO2. Okay. And again regeneration, through the regeneration mechanism, just there is a formation of PGA. So, that is why this photorespiration, there is no use of this photorespiration. So, one use, the CO2 will be produced in the form of PGA by this photorespiration mechanism. Okay. So, this photorespiration occurs in 3 types of cell organelles. Okay, photorespiration వల్ల ఏదైనా యూజ్ ఉందా అంటే ఎలాంటి యూజ్ లేదు C3 ప్లాంట్స్లో. ఏం జరిగింది ఇక్కడ మరి, మామూలుగా RUBP అయితే CO2తో బైండ్ కావాలి, అప్పుడే కార్బోహైడ్రేట్ సింథసిస్ జరుగుతుంది. కానీ ఇక్కడ RUBP దేంతో బైండ్ అయింది, ఆక్సిజన్తో, ఇన్ ద ప్రజెన్స్ ఆఫ్ లైట్. సో దట్స్ వై వి కాల్ యాజ్ ఫోటోరెస్పిరేషన్. బైండ్ అయినప్పుడు మనకి ఫస్ట్ ప్రోడక్ట్స్ ఏమొచ్చాయి, జనరల్గా అయితే PGA రావాలి. So, instead of the 2 molecules of PGA, then 2 molecules of phosphoglycolate వచ్చింది, phosphoglycolate. After the formation of phosphoglycolate, it undergoes rephosphorylation in the presence of phosphatase enzyme, then we will get the glycolate. Okay. So, this reaction occurs where, in chloroplast. After chloroplast, the second cell organelle is the peroxisome. In the peroxisome, there is an oxygenation. This glycolate undergoes oxygenation, then we will get the glyoxylate and H2O2. This H2O2 dissociates in the presence of catalase enzyme. Then this glyoxylate undergoes transamino